There was always critters in my backyard. A snappy turtle in my backyard, all kinds of snakes, other turtles, lizards, frogs, all of these different kinds of herpetofauna, reptiles and amphibians. And I was like, these guys are so cool. My dad and I would go fishing on what we called the finger canals in the Banana River all the time, almost every weekend. And we would walk along the riverbank and see what we could find, because there's always cool stuff just hanging around, washed. And one time we came across a horseshoe crab, and it was flipped upside down so you could see all of its arms and all of its appendages. And I didn't understand what I was looking at. What in its evolutionary history said, this is a good way to be. This is a functional evolutionary way to evolve. This is how I can go about life and be successful. I started volunteering at this place called the Enchanted Forest, which is a nature preserve in my hometown. When I was to find out more about reptiles and amphibians, I started getting into genetics. And the more I got into genetics, the more I was like, this is amazing. While I was in high school, I started taking Calc 2, and I met my Calc 2 teacher, who's really influential in my life. She's the one who made me realize that I can study genetics, but I need math to do it. So I can study math and genetics on whatever I want. And that realization was really powerful, very influential for my college career.
When I first started college, I knew that I had to get into something called research, and I wasn't sure what research was at the time, or like how it worked or what it involved. During my freshman year, I was looking through the UF website and the Florida Museum website, and I came across a section called herpetology, which is research in amphibians, which is what at the time I was really interested in studying. Started working with one of the grad students, and it ended up being a really good fit in terms of mentoring, my interest, us being able to work together. During this time, throughout the years, I went from starting only DNA extractions. For my first year, I did only DNA extractions and nothing else. And then I started working on the next steps. PCR, being able to send off samples for sequencing, getting those sequences back, checking them for quality. I reached the point where I started training incoming students. And these are all things that I got to do during undergrad, working in this lab. Being fortunate enough that the first lab I ended up in had such great mentors for me was very exciting, but also very critical to my journey through science. Because I know if it wasn't for them pushing me, I wouldn't have done a lot of the things that I ended up doing. So throughout college, I had planned on immediately starting my PhD and not taking any gap years in between, and not taking any time out of school, because I was concerned about my desire to go back kind of dwindling and wouldn't feel motivated anymore to continue my education. I first met Dr. Joe Ryan at the Southeastern Regional SACNAS Conference for 2020. I had given a presentation there, and I knew a little bit about his work from meeting one of his grad students previously. And I knew I was really interested in his work. He did a lot of evolutionary genomics, a lot of full-level genome analyses, gene family analyses, all this higher-level data that I hadn't yet worked with, but I knew I was interested in. And so afterwards, we ended up talking, and I learned that I can take a gap year, but still do research. So I can still learn, I can still gain more skills, I can still stay in this field that I found out I really love, but I don't have to be a student. So I can take time away from class and really just focus on my research and applying to grad school. And so that's how I ended up here at Whitney. Dr. Ryan hired me as a post-med, so a post-baccalaureate in his lab. He primarily works on the evolution of full genomes and looks at how developmental genes change between different animal lineages. And so while I've been here, I've been working on genome assemblies, so we can piece together the full genome before we start analyzing it. We have to piece together the full genome after sequencing, make sure the sequencing turned out well, make sure the genome makes sense, if you will, and then we can start looking at different evolutionary genes within them, and we primarily look at developmental genes. When you sequence a genome, which is all of the DNA in an animal, we can't sequence it all in one go. It's very, very large. We can sequence it in pieces, in small pieces, or in bigger pieces, and then you essentially have a file, just chunks of DNA. And so I come in, I take those chunks, and I start putting them back together, and it's almost like a jigsaw puzzle. Then we can start analyzing them. So that's where I go in, and I can look for different genes, and I can do what's called annotating those genes. So that's when I say this is where the gene starts, this is where the gene ends. These are the parts of the gene that are being expressed and being functional. These are the part of the genes that aren't actually used within the gene, and so those are removed. Once we have all of that information, we can really dig deep and start looking at how the gene works and how it's changed between different organisms. Since our lab is primarily a data analyst lab, one of the most important things we do is collaborate with other scientists who are the ones generating all of the novel data that we work on and that we then analyze and get back to them about. And we've been fortunate enough that here at the Whitney Lab, we can house visiting researchers while they do their research here. And we've had a visiting researcher in our lab, Dr. Dani, who's been here for the past several months. He's been generating novel data on this anemone nematostella that we can then start looking at how these different genes start working with each other. As my gap year comes to an end, I've realized I've learned quite a bit. If anyone ever asked me for a gap year, I highly recommend doing it. And I'm very glad that I was able to take time out of school and really prepare myself for my PhD. I was able to learn so many new skills, gain a lot more confidence in my coding skills and my analysis skills, being able to confidently know which programs to run and how to properly manipulate my files so the program can successfully run, and if it doesn't successfully run, figure out why. And for what I want to do for my PhD, working heavily in bioinformatics and genetics, this has been absolutely amazing for me. I was really able to take all of my new skills and my new knowledge and work them into my PhD applications and other scholarship applications. And I've been able to be really successful because of it. And of course, that's a lot of help from my former lab mates and my current lab mates. Absolutely wouldn't have been able to do it without them. And all of the hard work paid off. I've been accepted to Iowa State University to start my PhD. And as of August, I will officially be a first-year graduate student at Iowa State University.